Okay, well, without any further ado, I'd like to get us started here. I'm Laura Kolaskowski, and I'm one of the people with iConquerMS. I do have MS myself, and I've been with iConquer since the inception, and I'm really excited to be a part of this project of Chat with Chat. Just quickly, I just want to remind you all that iConquerMS, our mission is to partner with people who are affected by MS. Those are those of us who have MS, our care partners, our family members, researchers, scientists, funders, so that we can bring about changes for us, immediate changes. And we do that in several ways, and one of the most impactful ways we do it is through our research, and gathering research information from our participants is key to what we do. We have a couple different activities going on right now that I want to just remind you about. Real MS is our longitudinal study. It's research engagement about life with MS. And if you have not done this yet, please get online and complete your surveys. We do these every 6 months. Not only is it a good way for our researchers to track what's going on with us, it's an excellent record for ourselves to be able to use when we look back and say, what did I look like 4 years ago? What did I look like 3 years ago? It's excellent use of data. The other thing we have going on that's relevant to today is our study about COVID. And I'm excited about, we're going to be talking about that, but that COVID study is open. We're gathering data about people's experiences with COVID, with the vaccines, reactions, a wide variety of information's being collected. We also have our participant-powered research questions still in play, where we're actually evaluating the questions that are submitted for research, and we're going out and looking for researchers to match with those questions. So be sure to check out all those activities going on our website. Again, that's iConquerMS.org. There's our COVID-19 study, which we're going to be talking about today. And we also have some exciting initiatives going on. Our RIDE Council is now meeting regularly. It's a great group of people who really represent the diverse community of people with MS. Our pediatric network and our care partner initiative are gearing up and are also going to be live this year. So I am really excited where we're coming with this. But mostly today, I just want to introduce Chat, who is our host for this series. This is our 4th Chat with Chat. If you haven't viewed the first 3, they're available on our YouTube channel, and you'll get reminders in your email as to those links. And Chat will explain the concept of his Chat with Chat and introduce today's speaker. So Chat, I'm turning this over to you. And thank you everyone for joining us.
Well, thank you so much, Laura, and good afternoon, everyone. For those of you who is here for the first time, you might be wondering, right, why we name our webinar as a Chat with Chat. My name is Surachat. My last name, in fact, even longer, Ngo Suraches. But so I normally go with Chat. So when we have a group or our team kind of thinking about how are we going to put title together for this webinar, and then somebody just said that maybe we should call it Chat with Chat. So it took me a while to get used to the name, but right now I'm pretty good. So anyway, this is not all about me. So today we actually have a really interesting topic. So we are able to have our guest speaker, Dr. Farren Briggs. And I just like to take a moment to introduce him, and then let him talk about the COVID-19 vaccine safety. So Dr. Briggs got a PhD in epidemiology from the University of California, Berkeley in 2010. And currently he is an assistant professor at the Department of Population and Quantitative Health Sciences, School of Medicine at the Case Western Reserve University. And as I said, he's an epidemiologist, and he's actually his research emphasis is in the genetics and also environmental epidemiology. So he has various experiences investigating multiple phases of the etiology, pathology, manifestation of the chronic diseases, by design and also using diverse study designs, resources, and methodological approaches. And primarily, he's focused on the phenotyping multiple sclerosis at micro and to macro level, including profiling biomarkers, multi-morbidity, quality of life, physical, mental impairments, and so on. So that's so that is a little bit of the introduction of Dr. Briggs. And today he will spend like maybe 40 and 45 minutes with us talking about the COVID-19 vaccine safety. And it is a study from iConquerMS data. Please, Dr. Briggs.
Good afternoon, everyone. Hello from Cleveland, Ohio, where the sun is out, and I'm very happy. So I apologize for the poor lighting. So I'm very excited to present this work. I have been serving on the iConquerMS Scientific Advisory Committee for the last few years now, and I have a few other projects which I hope maybe in the future I might have the opportunity to present using Real MS. Just wanted to share back how impactful your participation is in the Real MS and other contributions to the field and knowledge gaps that we can address. But today I'm presenting work on behalf of the steering committee for the Cover MS project. This project, I will go through a brief overview of vaccines, as well as the study design, as well as our results. And feel free to ask a question. I won't be monitoring the chat, but Holly, maybe you can interrupt me as questions pop up that might be relevant in that time. But this is a very casual presentation. Feel free to reach out and we can have a conversation mid-presentation as well. So first thing is, why does it matter to study COVID-19 in MS? Well, these are results recently published, but we knew about them for a while from this large Italian cohort of individuals with MS seen at several clinics there. And essentially what they summarized was that individuals with MS had a twofold greater risk for adverse outcomes, including hospitalizations, ICU hospitalizations, being generally hospitalized, and unfortunately increased risk of death as well. So what this emphasizes is the need to really minimize SARS-CoV-2 exposure in persons with MS. What they were able to also do was really emphasize, well, who with MS are really at risk? And when they stratified by different subgroups, here on the left, we have a figure where they've stratified by individuals with a low EDSS score and no comorbidities, compared to those with a higher EDSS and at least one comorbidity. And you can see the results here. These are relative ratios. And when it is near 1, it means there's no difference. But here, these relative ratios are approaching 3, illustrating that individuals who had a bit more disability and had a few comorbidities were at higher risk for these adverse events specifically. So again, this emphasizes the need to urgently minimize exposure to SARS-CoV-2 in persons with MS, especially those with other comorbid conditions and those who have moderate to severe impairment. Fortunately, we are in the era where we have many vaccines. Here I'm highlighting just 4 of the most common vaccines available. We have the Pfizer, Moderna, Johnson & Johnson, and AstraZeneca. These represent 2 different mechanisms of action. 2 are mRNA delivery type of vaccines. The other 2 are using a viral vector. All of you are now familiar with generally the dosing strategies for these. And so with the mRNA vaccines, an mRNA sequence, which is essentially a blueprint of a DNA strand, is then injected to then allow our bodies itself to generate a protein that we can then develop an autoimmune, not an autoimmune, but an immune response to that will protect us from an actual exposure to the virus. With a viral vector, what happens is they use a non-replicating viral vector, another virus essentially, and they include a little piece of DNA from the COVID-19 virus, and that allows our body to recognize the COVID-19 DNA itself as an antigen. So these are the two approaches that are commonly used in the vaccines available here in the United States. So fortunately, these vaccines are pretty effective. They protect each of us, our families, and our communities, and both demonstrated to be really the two common ones, Pfizer and Moderna, both mRNA vaccines, demonstrated to be highly effective in preventing any symptoms related to COVID-19 in the clinical trials. This I just wanted to highlight in contrast to the flu vaccines, which are only about 40 to 60% effective every year. For that reason, this is why we need to develop a flu vaccine every year, because the flu mutates a lot. So as a result, we need to keep updating vaccines. As you all are aware now, we've gone through various strains of the SARS-CoV-2 virus. Most recently, we have dealt with Omicron. And so similarly, we might expect certain variation in effectiveness with various mutations of the SARS-CoV-2 virus in the future. But in general, these vaccines are pretty effective. They reduce any chance of hospitalization and death, and being unvaccinated increases your risk of really developing severe long-term complications. So a lot of questions people had was wondering whether or not the vaccine development was rushed. This is understandable. Within the blink of a year, we had a vaccine. I think everyone in the field, we were all pretty surprised. But I wanted to highlight a few key things here, was that all the vaccines have been developed by scientists and not governments. The vaccines were developed and tested quickly but safely, and none of the necessary steps were skipped in this process. The reason we were able to do so fast was two things in combination. One, there was a lot of funding. Lots of governments, lots of private foundations contributed money, and there was a lot of collaboration across countries, across companies. So the synergy of funding and cooperation is what accelerated the development of the many vaccines we have. And for most of these vaccines, the technology that was used actually has been in existence for many, many years. Here, I'd just like to highlight the general timeline that was underlying the development of the mRNA vaccines. This was sort of the first large-scale use of mRNA vaccines in the world, but this technology, the base of this sort of began about 30 years ago at least. The basis started with Dr. Karakio, who was developing sort of the technology for using mRNA technology. She then began collaborating with another colleague about how to successfully deliver mRNA, and this was in 2005, so this is 17 years ago. And then another team who began developing how to use this for a vaccine. This is 9 years ago. So again, this has been in development for a long time. And then in 2017, we had the first mRNA vaccine. This was for MERS, which is another respiratory virus. So this technology has been there. Fortunately, we were able to capitalize on this when the SARS-CoV-2 outbreak happened in 2020. And as a result, this is how we were able to really accelerate the development of a vaccine.
Farren, can I ask a question from our audience?
Yes.
One of our attendees wanted to know how long the capability for the RNA replication remains active in the human body after vaccination.
That's a great question. I think this was a concern for many individuals, right? Thinking about, you have this piece of blueprint of a DNA. It actually doesn't last that long. So I'm not sure of the actual time frame, but essentially the reason this is very appealing is that this degrades and it disappears within I'm willing to say in a matter of weeks. I'm not an expert here, but that's sort of the reason behind the desire to use mRNA. And in this situation, the viral vector is that both of these things are going to be broken down with a matter of time. So this is why they're very appealing. In general, that's the case for essentially all of the vaccine strategies, is that all of these components will be broken down. But an in general, as biologists, we think of RNA as being highly, not volatile, but it requires very specific conditions to be stable. So considering we're injecting this into a human being, I can only imagine that this may last. I can, at most, I would hypothesize a few days, if not a few weeks, but don't quote me on that. But again, this is why they're both very appealing, is that they disappear pretty fastly. Great question. So thinking of sort of the outline of a clinical trial has 4 phases. Phase 1, what we're really thinking about here is it's a small study. We're trying to think of, well, is a vaccine actually safe? Are there any severe side effects? That's generally the questions from this small phase 1. Phase 2, once you've demonstrated safety, you then ask the question, does it work? Can it elicit the biological response we're hoping for? This is in a few hundred individuals. And is there any evidence that it can work? And then we move into phase 3, which is your large scale studies to really assess how effective and efficacious are these vaccines in preventing disease. And these are the large scale studies that we all wait for to get these final results. So we have these multiple phases that happen. And then phase 4 is once a vaccine is now available for use in the general population, let's do some long-term surveillance to assess if there are any adverse events, any major risks, right? So this is the standard for pretty much all clinical trials. And so generally, just look at the top half of this figure. This is generally what it looks like in the normal situation. So we have a few years of, let's say, doing some exploration in animal models, in cell lines, looking at the literature, developing the hypotheses of how something might work. Then we have the safety assessment for a year. We then assess in a larger group of a few hundred individuals if there's any evidence that it might biologically work. And then we would do these larger scale studies for many more years. Then develop the infrastructure for large scale development of a vaccine, refining the manufacturing process, getting approval from the FDA, and then finally distribution. So we're looking at a decade-long process here. But this is what happened in COVID-19. What they did was they didn't skip any steps, but what they were able to do was begin doing some of these steps in the same time, in parallel. And because there was so much money in collaboration, they were able to really shorten the window. So instead of looking at a decade here, we looked at a year. It was amazing what we were able to achieve with such collaboration.
Farren, do you mind if I pop in with another question?
Sure.
Another attendee wanted to know, and I think this relates to the development process as well and who's included in these trials, but the question is, do those with autoimmune disorders have differing immune responses? And so how does that get addressed in this vaccine development, or is it an issue that companies would think about?
I will come back to that.
Okay.
I will hopefully address it in a few.
Great.
But great question. And so getting to this question, we all want to know, were people like me a part of any of these clinical trials? This is sort of the situation for the development of any drug, is that it's based on a clinical trial. And then me being me, I'm a multiracial person. I was like, well, was there a multiracial man in his 40s in this study using this drug? Is there any evidence it's going to work? Maybe there wasn't. So then it's going to be sort of best guess by the clinician. I am not a clinician, so I cannot provide medical advice, but that's generally how I approach something, is thinking, well, maybe there wasn't someone like me there. So what do I do? But here they did a good job of really getting people of all ages, diverse, racially and ethnically diverse populations, and individuals with various chronic conditions as well. So overall, what these large clinical trials were able to conclude were that the vaccines were generally safe and effective, and that they also provided immunity for individuals who already had a COVID-19 infection. But I'll come to the question on whether or not individuals with MS were participants in these trials. So looking at the racial breakdowns of the two largest two of the larger clinical trials. We see Moderna had a high representation of diverse populations, less so for individuals who were American Indian or Pacific Islander. And similarly for Pfizer, had a large Hispanic and Black population, sort of representative of the distribution of these groups in the larger general US, in the general US population itself. So here is the overall effectiveness from these two large studies. If we look at this one figure, essentially this is a representation of new infections of COVID-19. So those who received the vaccine, the Moderna vaccine, very few new infections occurred, very few. The line basically stays flat. But in the individuals who received the placebo, many more, right? So they were able to calculate that effectiveness of the mRNA vaccine over the duration of the trial was 94%, which is pretty impressive. When they're able to divide the data based on people of color or not, we could see that the vaccine was 97% in people of color. Similarly, for the Pfizer vaccine, here is the Pfizer vaccine. We see very few new infections on those who are vaccinated during the period of the clinical trial, which is here about 3 months, over a 3-month window. And here are the new infections of those who were on the placebo. Overall, the Pfizer vaccine was 95% effective. And in the 10% of individuals who are Black, it was 100% effective. And in the 28% of individuals who are Hispanic, it was 94% effective. So in the large-scale clinical trials, by race, these vaccines were also very effective. So there's no variation by sex or race, and nor age as well. So again, very promising and strong results. This is why things moved forward pretty fast, is that these data were pretty robust and pretty surprising to all of us at how effective they were working within these 3-month windows. The question then comes up from all of these clinical trials, well, what are the side effects? And most of the side effects reported were mild, generally occurring within a few days. And as many of us have anecdotally heard and might have experienced ourselves, was that these side effects were also stronger after the second dose for some individuals. The most common included pain, muscle ache, fatigue, headache, nausea, but there were also other symptoms as well, but they were less common. And there's a typo here, and serious side effects were very rare across all of the clinical trials. And interestingly, at least from their data, it showed that the vaccines were also safe in those who had allergies of various kinds.
Farren, I've got another question. You mentioned chronic conditions or chronic issues they were looked at, and the vaccine seemed to be effective in people with those. Can you just define what you mean by chronic? Like what kind of conditions were included?
Highly variable what I mean by that, because each of the clinical trials had different inclusion and exclusion criteria. So generally, they included individuals who had conditions that did not require immunosuppressive therapy, individuals who are not immunocompromised. They excluded individuals who had cancers, most of them, or HIV, who were HIV positive. And I'll show in the next slide a little bit more of how they approached MS. But these were individuals with, I would say, well-managed chronic conditions. You can go through all of the protocols, look at the inclusion-exclusion criteria. So it's individuals who had well-managed or stable health, essentially, were included. And there was no real stratification of the results by the various comorbidities in the original publications, but I think others have started to look at that, and we're starting to see, it seems that the vaccine still performs as well in individuals with stable chronic conditions who were recruited as part of the clinical trial. But again, good question.
Holly, there's another question. Would you like to take that?
I will come back to that question. Let's try to revisit that question a little bit later on in today's presentation.
Sounds good.
So these side effects basically for the vaccines were considered to be fairly mild. So how do these side effects compare to other vaccines out there? Here we have Moderna and Pfizer. Here we have the quadrivalent flu vaccine. Here we have the shingles vaccine. And so here is the percentage of individuals who experience a side effect, but this is in the group who has the most side effects. So it was shown in all of these clinical trials that the clinical trials for the COVID-19 vaccine that younger adults had more side effects. So under the age of 55, they had more side effects, as well as women. So women and under the age of 55 in all of the major clinical trials showed to have had more side effects. And so in this high risk population for side effects, here are how these other vaccines perform. About 46% of individuals in this high risk group experienced some side effect. To Moderna, 36 to Pfizer. To the flu, on average, they experienced only 15%, but for shingles, 48%. So, you know, the COVID vaccine side effects is right in the middle of these other commonly available vaccines. Again, the most common side effects in the general population in this high-risk group is local pain, fatigue, muscle pain, headaches, and so forth. Here is severe side effects, which I call overall grade 3. And here it's 5% who was on shingles, 4% with Moderna, 1.5% with Pfizer, and 0.5% with the flu. Again, this is in this younger group. But again, this is sort of reassuring in illustrating that these vaccines, these new vaccines, are performing comparably to other existing vaccines that we've studied and had around for a very long time. So coming to that other question, were people with MS a part of these clinical trials? Well, no. Pfizer excluded individuals with a history of autoimmune disease, including MS, and I'm quoting directly from their protocols. Moderna excluded individuals who received systemic immunosuppressants or immune modifying therapy for 14 days in a total of 6 months prior screening. They did not specifically say MS in their exclusion criteria, but we can assume that individuals on immune modifying therapy within the for at least 14 days and 6 months prior were excluded. So many individuals. There's no details on autoimmune diseases specifically, so I'm willing to say that they were excluded because none of that information was reported. For Johnson & Johnson, they excluded individuals with clinical conditions, for example, autoimmune diseases, which were expected to impact immune response. They explicitly stated why they excluded individuals with autoimmune diseases. And then AstraZeneca excluded individuals with any autoimmune condition not requiring immunosuppressive or immunomodulatory therapy. So again, we can assume this included all individuals with MS as well. So generally, since the granular data have not been provided as such, we can only assume that individuals with MS and pretty much most other autoimmune diseases were excluded from all of these clinical trials. So this sort of ties into vaccine hesitancy in individuals with MS. You know, if individuals with MS are not a part of these clinical trials, you know, many questions can arise. First, thinking about, well, is the vaccine safe for me? Does the vaccine vary in effectiveness given I have an autoimmune disease? What's the relationship with DMTs? All of these questions that many of you submitted beforehand, and as a result, the scientific community has tried to rally to address many of these questions. So coupling with the fact of this lack of information in the clinical trials for individuals with MS is also vaccine hesitancy. So within the United States, there's this recent paper, there have been several studies looking at vaccine hesitancy across the world, and this recent paper does a nice job of summarizing vaccine hesitancy across the many studies that have looked at this. And 22% of persons with MS in the United States are vaccine hesitant, specifically related to the SARS-CoV-2 vaccine. This is a concern considering the risk for severe adverse events that I showed you in the first 2 slides. There were many concerns that tied to hesitancy, including the lack of information, not wanting to change one's lifestyle, religious concerns, so forth. But one of the major reasons for being vaccine hesitant was people were unsure about short-term side effects as well as long-term. These are both major concerns. So it's a major concern for 10% of all persons with MS and 30% of those who are vaccine hesitant. So again, here are knowledge gaps that we could address. And the reason we have this knowledge gap is because people with MS were not a part of those clinical trials. And so this has sort of led to this study here today. iConquerMS has the portal which Laura shared earlier. Questions have power. And through the Questions Have Power portal, which individuals with MS and participants in iConquer submitted various questions they had in COVID-19, along with the steering committee, tried to figure out, well, which questions can we answer rapid and that could have an impact? And so iConquerMS and the steering committee were trying to think, how can we help rapidly? And one of the first questions we came up with that we could address was that, can we answer vaccines are safe for persons with MS? Just that question alone can hopefully mitigate concerns around vaccine hesitancy for many individuals. And that can hopefully encourage individuals to be vaccinated, and just by demonstrating that the vaccines are safe. So this led to iConquerMS developing the COVER-MS study, which was funded by the National MS Society. And I just wanted to show you in this figure what a quick response this was, right? Here we have the start of the pandemic, January of 2020. The world shuts down for the most part in April of 2020. We see vaccines being developed in December of 2021. And around here, the team, Holly, Sarah, and the rest of the team were managing the Questions Have Power. The steering committee for COVER-MS met, and we were able to come up with the study design to address some of these questions. And it was able to launch so fast by March of 2021. So this was a rapid response about how can we generate information to fill in some of these key gaps. And I'm very, very proud of how fast we were able to launch this study. It took a lot of hard work on the back end by the iConquerMS team, and it has resulted in some meaningful work and that I look forward to sharing with you in the next coming slides. So you all know iConquerMS. And so this study was launched in March 2020. And one of the key questions was, what were the short-term side effects experienced by individuals with MS in response to the vaccine? Other information has also been collected, and we are going to be looking at this in our future analyses. We collected information on relapses, symptoms, as well as long-term reactions, as well as response to reactions to the first, second, and the any booster someone has had. So this is ongoing work. And as of today, I checked this morning, so far, 1,533 individuals with MS have enrolled. And if you have not joined, do so. You can click on this link. On this link, there's also a link to the same results that I'm presenting here today, as well as a very interactive dashboard for you to see results, general descriptive results of participants who have participated so far in COVER-MS. So brief overview, I'll describe the participants in COVER-MS, this first analysis that we have done and published. And then I will show you some data illustrating vaccine reactions in the COVER-MS population to the first vaccine and the second vaccine dose, if applicable. Then show you some data illustrating the severity of vaccine reactions in the COVER-MS participants, and then showing you the severity of COVER-MS, the severity of vaccine reactions in COVER-MS by DMT status and age. And the last one I'm not showing here, I'll show you a final set of results about what are the independent predictors of vaccine reaction, vaccine side effects. So for this analysis, we waited 3 months, and within 3 months, 719 individuals had reported having at least one vaccine, and 442 had received their second vaccine within this, so within a 3-month period. And this is where we froze the data to do the first round of analyses. As expected, there was a majority women. Mean age of life was around age 55. Majority of individuals had relapsing remitting, but we also had secondary progressive, primary progressive. And these distributions are as expected for the general MS population. And here was this breakdown by types of vaccine. Most had received the Pfizer. A large percentage had Moderna, but also we had Johnson & Johnson, AstraZeneca, and other. Individuals from across the world were able to participate. But for the sake of this analysis, we focus only on AstraZeneca, Johnson & Johnson, Moderna, and Pfizer. So the first question is, how do the short-term vaccine side effects in persons with MS compare to those reported in the clinical trials? Well, here in blue, I have the data from the clinical trials, the 4 major clinical trials averaged, and then I have the results here from COVER-MS. And what you can see, well, first is that COVER-MS participants reported less pain than those in the clinical trial. However, COVER-MS reported more fever than the clinical trials. So one might ask, well, does that mean people with MS experience fewer side effects? Well, one of the driving factors could be age. The clinical trial included many more younger adults, and in younger adults, they experienced more side effects compared to older adults. The population for the COVER-MS study was around, the mean age was around age 55. So it was slightly older. So this could be driven by age, but overall, here are the reactions after the first vaccine. Nothing glaring is popping up. So this is really reassuring. Here are the reactions after the second shot. Again, things jump up now. They're a little bit higher, but again, it is lower for the COVER-MS population across all of these various symptoms, but again, a little bit higher for fever. Again, age could be explaining this pattern here also, but what's reassuring again is there's no dramatic red flag here to suggest that we should be concerned that there are major side effects to the vaccines in persons with MS. So here's taking those 2 figures before and combining them together. And you can just see here is the RCT data, which is in blue and orange yellow for COVER-MS. And we can see that there is this nice trend that there are no majorly different factors, except maybe greater experiences of fever is the only thing that's sort of more common in persons with MS. So concluding that is overall persons with MS did not experience more short-term vaccine side effects than the non-MS population in the clinical trials. So question 2, what were the most common side effects of persons with MS and how severe were they? Here are the reactions after the first vaccine. The darker color is more severe. And so about 55% of individuals reported pain and 5% reported severe pain. The most severe symptom experienced by most were fatigue. 11% of the participants reported severe fatigue after the first shot, but otherwise these trends were pretty similar to everything we saw in the general clinical trials.
Farren, there is a question, I think, comparing to the clinical trials data where people with MS had maybe less pain or fatigue than the trial participants. The question is, since pain and fatigue are normal for people with MS, could it just be that people didn't see a change in the vaccine because these were things they experienced on a regular basis anyway?
Yeah, good question. I think, well, part of the COVER-MS, the questions were specific to the vaccine, right? So within the 24 hours after the vaccine, did you experience an increase in pain? And so I would think participants would have responded in relation to the vaccine because there were also separate questions about your MS symptoms at that point. Which is a good point, and I think we should look at this, we should compare current symptoms to side effects experience to really refine that point. But I do think these were in response to specifically the vaccine. And Holly, you could probably speak to that some more as well.
I don't think I can. I think you answered the question really well, but I think you're right. We could go and look and see what people were reporting in terms of their MS symptoms and see if there's any correlation. And then there was a similar question from another person about, again, MS patients having already chronic pain or less sensation. It could be that they have pain or they don't have as much sensation, and that could affect the pain responses. And then another person says that many people are already on regular pain medication. So there's a lot of great reasons why we might have seen that.
Yeah, yeah. Which I think in conclusion for me though, what's the good takeaway was that there wasn't an exaggeration of any of these symptoms except for fever. So which should lend the idea that the vaccine should be generally safe. At least it was safe in this population. Those who participated by this time point, it appears the vaccine did not have exaggerated short-term side effects, right? So, and I think those are really great points that could explain these various patterns that we see here as well, and things that could be looked into in the future. I do like these ideas a lot. A colleague of mine, a mentor of mine says, good science always begets more questions. And so the more questions, it just gets more questions, because then we want to know why, right? Knowing why is very important. And so going to question 2, we're looking at which symptoms were more severe. And this is after the second shot. You can see how things increase overall. So we go from 55% with pain, and let's say about 15 with fever, to after the second shot, we have 65 now and about 30%. So we have a doubling of individuals experiencing fever after the second shot. My personal experience was that I had no side effects after the first shot. I'm not a person with MS, but I had no side effects after the first shot. After the second shot, I was knocked out for the longest period of time. So I'm glad that I was potentially an outlier, having really severe headache, fatigue, and muscle aches. But I was one of these outliers, and it could be for many reasons or not. It just means that I was unlucky in that way. But again, we don't see anything major. All of this is pretty consistent with the literature, and so overall, the side effects were mild to moderate. The common side effects were pain at the injection site, fatigue, headache, muscle aches, and malaise. And side effects were more common after the second injection. Again, these were all consistent with what we've seen in the larger studies, the clinical trials. So then what were the patterns of these side effects by DMT status and age? Here we have the data just generally dividing by any DMT or no DMT. And individuals with any DMT were experiencing a bit more, report a bit more pain, but there was a little bit more elevation of headache, maybe malaise, but again, the patterns were pretty consistent with the exception of pain and this is pain at injection site itself. Here it is after the second shot. We see an increase generally across the board for people on any DMT versus no DMT. Again, things that could explain this could be age, where older adults in the population may not be in a DMT, and that could explain this. But again, we were unsure, but this was, this was curious findings that we wanted to explore further, which I'll show you in a few more slides. And so we had an increase in pain, fatigue, and headaches here for the NE-DMT group. When we look at it by age now, you can see the oldest age group had fewer side effects across the board compared to those who were in the middle, age 40 to 60, and then those who were the youngest group, they had even higher experiences of pain, but not too different from the mid-age group here. Here it is after the second shot. Now you can see that it really is exaggerated for the younger age group. They are experiencing more pain, fatigue. I mean, 85% of persons with MS under the age of 40 reported at least pain at the injection site. Just about 75% reported fatigue. And this is in comparison to only 40% of the older adults in the study, right? So age is a huge driver of experiencing a side effect in the MS population. So the takeaways from here would be Persons with MS on DMTs may be more likely to report slightly more moderate pain, fatigue, and headaches. And those under the age of 60 were more likely to report moderate side effects, especially after the second shot for those who were under the age of 40. So then the last question we have here is, what were the independently associated factors with experiencing a side effect? So in the prior slides, I showed that age could be related, DMT status could be related. So we really want to now figure out, well, let's now adjust for all of these potential other factors to see which ones are actually driving these associations that we are observing here in these histograms. And we did this by doing a multivariable statistical regression model. Essentially, we're taking all of the variables and we're putting them into a model to identify which ones are the most important drivers of experiencing side effects. So this is a big and busy table. I just wanted to summarize the results really quickly. We had 2 outcomes. This is experiencing any reaction after the first shot. This is experiencing any severe reaction. And so here, what I'm showing you are odds ratios, which are essentially relative probabilities. And an odds ratio of 1 means no association. An odds ratio less than 1 means it's protective. So for example, here, for increasing age, the odds of experiencing a reaction decreased. It's less than 1. So older age is protective. If it's greater than 1, that means you're experiencing more symptoms. So here, females had an 89% increased odds of experiencing a side effect. This is accounting for all of these variables. So we put all of these variables into the model. We saw a slightly protective effect for being non-white, but this did not persist across a severe group. But essentially, what we can summarize is that factors associated with greater risk factors were younger age, being female, having a prior COVID-19 infection. Here is that association. You were 3 times more likely to experience a side effect if you had a COVID-19 infection. And individuals who were on AstraZeneca, which is here, you were 6 times more likely to experience a side effect compared to Pfizer. What's notable were factors that did not make any difference. There was no difference by MS subtype, disease duration, nor disability. Here for PDSS, maybe something suggestive for severe reaction, but again, this generally, there doesn't seem to be a pattern. There were no differences generally by race, no major differences. If anything, non-white individuals were less likely to report a severe reaction. And then there were no major patterns by DMT status. So all of these comparisons for various DMTs are in comparison to not having a DMT. And so there were no significant patterns here suggesting that specific DMTs were associated with more side effects. If anything, individuals on S1P modulators and alpha-4 integrin blockers, so Gilenya, Myzant, Zeposia, and Tesabri, were less likely to report side effects. So if anything, they had fewer side effects if you were on specific DMTs. But in general, there were no differences by DMT. And there were generally no differences between Moderna and Pfizer in terms of experiencing side effects. Here it is for after the second shot. Major factors, we see the common theme again. Younger age is associated with more side effects. If you had a side effect after the first shot, you were more likely to have a side effect after the second shot. You were 12 times more likely, which is really strong association. People who had Moderna were also more likely. They were 2.6 times more likely to experience a reaction after the second shot compared to Pfizer. But again, major things here were that at this point now, sex did not differ. Women did not experience more side effects after second shot. There is no real difference here by race or ethnicity. There is no difference by MS subtype, by disease duration, by disease severity. And most DMTs showed no relationship. Again, the only ones that showed some evidence of less severe reactions were those on the the S1P receptor modulators, and the alpha-4 integrin blockers. So again, here are two groups, I'm sorry, only this one group was experienced fewer symptoms, and maybe people who were on Tecfidera and Vumerity, the fumarids, were experiencing fewer. So again, no evidence that people on specific DMTs were experiencing more side effects. And again, at this point, prior infection played no role in experiencing side effects after the second shot. So now we have this clearer picture here about what's truly driving the association, right? So it's younger age and maybe having received a specific vaccine type. And if you have a proclivity for experiencing a side effect after the first shot. So takeaways from this would be being younger, female, and a prior COVID-19 infection were more likely to experience short-term side effects after the first vaccine shot. Being younger, reporting a side effect after the first shot, were more likely to experience short-term side effects after the second shot. We saw no major differences by MS attributes such as subtype or disease duration. And we saw no differences for the majority of DMTs, with a few suggesting fewer side effects, if anything, but none illustrating more or a higher burden of side effects.
Farren, we've got another question about whether there's any effect on MS relapses. And I don't think it's something we've looked at yet, but we can. But have you seen anything in the literature, or do you have any personal thoughts on that?
We were just at the American Academy of Neurology meeting in Seattle last week, and there were several presentations on COVID-19 and disease activity. And I will say there's not a lot out there yet. I think it's still unclear the impact of COVID-19 on MS. It doesn't seem, no one has looked at the vaccine itself on MS symptoms or side effects on MS symptoms, but they have looked at actually having an infection. And it seems individuals with an infection may be more prone to long-term COVID-19 symptoms. That was one of the studies I saw. But in terms of side effects, there's no evidence of a relationship between side effects and MS symptoms or relapse activity. And that's something we are hoping to do in the future, the next component of the COVER-MS study here. Okay, so recapping big picture, persons with MS do not have more reactions to the vaccines than a general population. Most of these are mild. Those on various DMTs, when we now adjust for age and all of the other potential variables that could differ, there were no major differences suggesting any adverse events for specific DMTs. There were no differences in subtype and disease duration as well. Older adults and men reported fewer reactions. Those with a prior infection of COVID-19 reported more reactions. And if you had a reaction after the first shot, well, it was likely you were going to have a reaction after the second shot. And this work was published September of last year, so pretty fast. We launched the study in March. We did the first data freeze in June. I worked with the team and did all of the analyses in July. We wrote up the paper in August. We submitted it and it was accepted pretty fast, right? This was an unusual accelerated process, but I think everyone understands the urgency for doing such work. So it was a very positive experience for all of us involved. So I just wanted to highlight for those of you, here's the current status of the COVER-MS. This is data, I just took this off of the dashboard. So far, there are about 1,200 individuals who report their first vaccine, 800 who report having a second, and just under 300 who report having had at least one booster. And here are the actual counts of individuals reporting a vaccine reaction after the first, second, and third dose. And right now, things are looking on track that there aren't any major, there's no major evidence, just glossing, just look at first glance, that there are no major evidence for additional side effects once you've had the booster. So we're looking here at about 15% of the 270 are reporting any pain. About 10% are reporting fatigue. And so it's very similar numbers to what we saw for the first and second vaccine dose. So with this, This project was only possible through the leadership at iConquerMS and the steering committee, specifically the PI, who was Robert McBurney, the co-chair, Phyllis Klein, and then the many members who included a few researchers, a few clinicians, and several individuals with MS as well were part of the steering committee to sort of guide how we conducted the study and how we interpreted the results as well. This work was made possible through funding from the National MS Society and specifically those who chose to participate, really demonstrating the capacity for really rapid research that can have a positive impact on people with MS. And with that, I'd like to conclude and happy to take any additional questions at this point.
There was just one more question about any people with MS. Could we identify whether there's anybody diagnosed as a long hauler, so somebody with COVID-19, long COVID, and that's another thing we'll be looking at in our next go round of CoverMS. So
yeah, I think that's sort of information we are hoping to now design as part of the next stage of CoverMS. So, you know, we asked this first question of, is the vaccine safe in the short term? And I feel pretty comfortable and confident saying, yes, it is. Now we have to then ask questions, are there any relationships with MS symptoms? Are there any relationships between side effects and symptoms? Thinking of prior infection. So again, it begets many more questions, and I think this is why we need to stay engaged here. And this is also why the questions have power really exemplifies this unique opportunity of tackling questions that really matter, particularly to the MS community, driven by people with MS. And I'm super excited by how quickly we're able to sort of turn this around, and I look forward to the work we can do with the CoverMS project and all of your participation.
All right, I just look at the clock right now. It's 4 o'clock, and I'm pretty sure that you know, people still have more questions, and I strongly believe that Dr. Briggs are more. I mean, those questions would be more welcome, and you know, you can also either send it to iConquerMS, or in fact, you know, you got the email from on your screen right now. You can actually send the question to Dr. Briggs as well. I am pretty sure that you will get a response from him. And I just would like to take this opportunity to thank Dr. Briggs for being with us today. It's very informative, very interesting presentation, and you can see you know, a few people give compliment in the chat function as well. So thank you so much, Dr. Briggs. And just a quick announcement. As I said, this is I did not really mention that this is the first time of the webinar. I mean, the first webinar for this year in 2022. So we will have the second one on June 8th at 4 p.m. Eastern Time. And we have Dr. Wallen from VA Medical Center, and he will talk about the telemedicine. So please stay tuned, and I believe that you will hear more details from iConquerMS, either websites or Twitter or all other social media. Again, thank you so much, everyone, for being here today. Thank you. Bye.